In this video, we will discuss about pancreatic cancer TME design. So, the previous video we have understood that this paper discusses about this oncolytic herpes simplex virus, how it can be used to make the tumor microenvironment of this pancreatic cancer more or less immunosuppressive, so that it can be easily detected by the immune cells, and immune cells can act against the pancreatic cancers. So in order to check their hypothesis, they need to have a system where they can have the TME that is close to the in vivo TME that people have observed. So for that, they designed this TME. So first they checked the immunofluorescence. First they checked the markers for this pancreatic stellate cells using immunofluorescence. So they checked the alpha smooth muscles, and they also check this FAP, that is fibroblast activation protein. And both of them are present in these pancreatic stellate cells, so which is also expected, as these are the markers for the pancreatic stellate cells. After that, they checked in the flow cytometer. The flow cytometer, there's these two things, it is fibroblast activation protein and alpha smooth muscles in these pancreatic stellate cells and also in the cancer associated fibroblast, and they do not see a significant difference in both the cases. So in this case, they are able to check this FAP and Alpha-SMA in two different conditions, that is pancreatic stellate cells and the cancer associated fibroblast. Now, in order to make the model, what they decided to use the two different cells that are injected into the mice at 1:10, that is KPC. KPC by PSC is 1:10, and that mixture they inoculated the mice, and then once the tumor reaches to this 1000 millimeter cube, they take out the tumors and analyze these tumors using flow cytometry and Sirus staining, and they checked the signals for the FAP, that is fibroblast activation protein, which is significantly higher in the 1:10 mixture compared to the control case, which is the KPC mixture. And they also did the Sirus staining to check the signals for this collagen, and they are able to see the high amount of collagen in 1:10 mixture compared to the control case. And these things they have also quantified in the panel H, where we can see that the collagen in the 1:10 mixture is significantly higher compared to the control. Now in this model, they are able to see high collagen. At the same time, they are able to see this high FAP productions, and which is also in line with what people have observed in the in vivo tumor microenvironment. So far from these results, they just made a TME that can be used to check the effect of this oncolytic herpes simplex virus, as we have discussed in the previous video. That is oncolytic herpes virus. They are checking the effect of this oncolytic herpes simplex virus on this TME, and what are the things that it actually changes. So, and in the abstract, they have mentioned that they have seen the changes in terms of this upregulation of this antigen presenting cancer associated fibroblast and downregulation of this myofibroblast, which is immunosuppressive in nature. So far they have not come up to that point in this first figure, and this is also not the completed first figure. We will discuss the other part of this first figure in our next videos. Here they have only checked whether the 1:10 will provide 1:10 inoculation of this KPC to pancreatic stellate cells will provide TME that is more in line matches with this compositions that are observed in the in vivo tumor microenvironment of pancreatic cancer cells, that is high FA, high CA, high collagen formations, and high fibroblast activation protein formations. I hope you understood their pancreatic cancer TME design and how they validate their pancreatic cancer TME design, that is using checking the markers, that is FAP and collagen. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time. I have discussed some things at least 2 times in this figure so that people are able to understand what I am trying to convey or what this paper is trying to convey. So again, thank you for your time.